ever wondered how traits get passed down from one generation to the next? You know, why you might have your mom's eyes or your dad's hair color. This is one of the biggest questions in all of biology, a puzzle that people observed for thousands of years before we finally started to understand the hidden instructions written inside every single living cell.
It seems like a ridiculously simple question, right? An elephant has a baby elephant, a mango seed grows into a mango tree. We kind of take this for granted, but behind it all is the profound science of heredity. So how does life pull off this incredible continuity while also allowing for all the amazing variations that make each one of us unique?
To really get this, we have to go back in time. For thousands of years, our ancestors were actually brilliant, practical geneticists. They knew that if they bred certain plants or animals, they could get the traits they wanted. It's how we ended up with things like high-yield crops and domesticated animals like the Sahiwal cows in Punjab. They knew what worked, but they had absolutely no idea why. The actual mechanism behind it all was a complete black box.
Okay, so before we go any further, let's nail down a couple of key terms. First, you've got inheritance. That's the actual process of traits being handed down. But just as important is variation, which is the reason you're not a perfect copy of your parents. The science that dives into both of these ideas, the science that pries open that black box, well, that's what we call genetics.
The very first person to really shine a light inside that box was a bit of an unlikely hero, a 19th century monk named Gregor Mendel. For 7 years, between 1856 and 1863, he ran thousands of super careful experiments on simple garden pea plants. And in doing so, he pretty much single-handedly laid the foundation for the entire field of genetics.
So what was Mendel's secret sauce? It was all about his methodical, almost obsessive process. He picked the perfect test subject, the pea plant, which had simple, easy-to-track traits. But the real game changer was this last part. He counted everything. See, at a time when biology was mostly just about describing what you saw, Mendel brought math and stats to the party. This was absolutely revolutionary. It turned the study of heredity from just guesswork into a real predictive science.
All right, let's walk through his most famous experiment. He decided to keep things simple and just look at one trait at a time. In this case, how tall the plant was. So he took a plant that he knew only ever made tall offspring and crossed it with one that only ever made dwarf offspring. The big question was, what happens when you mix them? And the result in this first generation of baby plants, what scientists call the F1 generation, was honestly pretty weird. Every single plant was tall. The dwarf trait didn't get watered down or anything, it just vanished completely. It wasn't a mix, not a medium-sized plant, just tall. So where on earth did the dwarfness go? This mystery led Mendel to his first huge insight. He figured that for every trait, a plant gets 2 sets of instructions, one from each parent. And sometimes one of those instructions is just louder than the other. This louder instruction, the dominant one, is the only one you actually see. The quiet one is still there, it's just hidden. He realized that tallness was the loud instruction and dwarfness was the quiet or recessive one. But Mendel was a great scientist, so he didn't stop there. He took these tall F1 plants and let them self-pollinate. And in the next generation, the F2, that hidden trait came roaring back. The dwarf trait was back. And get this, it wasn't random. He counted them all up, and the numbers were undeniable. For every 3 tall plants, there was exactly 1 dwarf plant. A perfect 3-to-1 ratio. How crazy is that? So how in the world do you get such a precise 3-to-1 ratio? Well, this simple little chart, called a Punnett square, is the key. Remember those instructions Mendel talked about? We call them alleles now. So the tall allele gets a capital T, and the recessive dwarf allele gets a lowercase t. Those first-generation plants had one of each, a TT genetic makeup. When these plants make their reproductive cells, half get the big T and half get the little t. This square just shows us all the possible ways they can combine. You can get 1 TT, which is tall, 2 TTs, also tall because T is dominant, and 1 TT, that's our dwarf. And there it is, 3 tall to 1 dwarf. It's just a beautifully simple explanation for a really complex result. And just from this one set of experiments, Mendel figured out 2 of the most fundamental laws in all of biology. First, the law of dominance, which explains why that dwarf trait disappeared at first. And second, the law of segregation. Now this one is so important. It says that those 2 alleles, T and T, they don't blend. They stay separate and are passed on independently, just waiting for a chance to reappear in a future generation. Now, Mendel's laws were elegant, they were powerful, and for a long time, it seemed like they explained everything. But you know how science is. As soon as you think you have a rule, you find exceptions. Scientists started looking at other traits in other creatures, and they found these fascinating cases where the simple rules of dominance just didn't seem to apply, showing us that the story of inheritance was even more complex than Mendel ever imagined. Here's a great little summary of what they started to find. Over here, you've got Mendel's classic dominance, where one trait just completely takes over. But then you get these other cooler possibilities, like what if neither trait wins? In incomplete dominance, you get a blend, something totally new in the middle. And then there's codominance, which is like a tie. Both traits show up at the exact same time, both fully expressed. A perfect example of incomplete dominance is the snapdragon flower. You cross a red one with a white one, and you don't get red offspring, like Mendel's rules might make you think. You get pink. How cool is that? This happens because the red allele just isn't strong enough to totally block out the white one, so they kind of meet in the middle and you get this beautiful blended look. Now for codominance, we can actually look at our own bodies, specifically our ABO blood groups. The instructions for A-type blood and B-type blood are equally powerful. So if you inherit one of each, you don't get blend. You get type AB blood, where your cells are literally making both A and B markers. This example also shows us something else cool. Sometimes there are more than just two versions of a gene out there. For blood type, there are three main alleles in the population, A, B, and the recessive I allele, which gives you type O blood. So Mendel had figured out these incredible mathematical rules for inheritance, but his factors were still just these abstract concepts. Nobody knew what they were physically made of or where they were hiding inside a cell. The next giant leap in genetics was all about finding that physical proof. And this timeline tells such an amazing story about science. Mendel published his work way back in 1865 and nothing. Crickets. For 35 years, it was basically forgotten. But then, right at the turn of the century, everything changed. And you know why? Technology. Microscopes had finally gotten powerful enough for scientists to watch cells divide, and they started noticing these strange thread-like things moving around in very specific, very predictable ways. And this was the big aha moment. In 1902, two scientists, Walter Sutton and Theodore Baveri, working totally independently, noticed that the way these threads, these chromosomes, behaved during cell division was a perfect one-to-one match from Mendel's abstract laws. Think about it. They both come in pairs, the pairs split up when sex cells are formed, and different pairs sort themselves out independently. The conclusion was just inescapable. Mendel's factors had to be on the chromosomes. This powerful idea was officially named the chromosomal theory of inheritance, and for the very first time, the abstract rules of genetics were connected to a physical thing that scientists could actually see and study. This didn't just prove Mendel was right, it completely blew the doors open for modern genetics. And this whole journey, from a monk counting peas in a garden to scientists staring through microscopes, it's not just some cool history lesson. This fundamental understanding of how inheritance actually works is the engine that drives so much of 21st century science, from medicine to agriculture and way beyond. And of course, the story kept going. Scientists like Thomas Hunt Morgan, using tiny little fruit flies, not only proved the chromosomal theory was spot on, but they added new layers of complexity. They discovered that genes sitting close together on the same chromosome tend to travel together as a package deal, a concept they called linkage. This discovery was the first step toward actually creating maps of the genes themselves, opening up a whole new world in our ability to read the book of life. I mean, it all started with a simple question about why some pea plants were tall and others were short, and from that, Gregor Mendel unlocked the first fundamental secrets of the code that writes the story for every living thing on this planet. His work kicked off a chain of discovery that is still accelerating today, from mapping the human genome to literally editing our DNA. And it just leaves us with this incredible, inspiring question: what secrets are we going to unlock next?